Welcome everybody. If you've already joined us, we're going to take a couple of minutes while we wait for everyone else to log in, and then we'll get started very shortly. We are at the 2 o'clock mark. We will give it just a couple more seconds to allow people to trickle in, and then we'll get started. Okay, welcome everybody, and thank you for joining us here today. We're excited to have everyone here at our webinar on the characterization and selection of antibodies for COVID-19 diagnostics using surface plasmon resonance. My name is Sajni, and I'm an application scientist here at Nicoya, and I will be your moderator today. Before we begin, I'd like to take a moment to cover just a couple of housekeeping items. So the webinar is being recorded and will be made available to all attendees after the event. And throughout the presentation, if you do have any questions for our speakers or for the topic at hand, please do send them to the Q&A chat box at the bottom of your screen, and we will be sure to address all questions during the live Q&A period at the end of the session. So it's my pleasure to introduce our 3 speakers today. Please join me in welcoming Soleil Grise, Senior Product Manager at Nicoya, Dr. Michael Piazza, Systems Integration Manager at Nicoya, and Dr. Rob Burgess, Chief Business Officer at Sinobiological. As Senior Product Manager, Soleil supports the development of innovative surface plasmon platforms, having pursued a BSc from the University of Waterloo in Canada, as well as her CFA Level 1. She leverages her background in science and technology with her passion for technology adoption as she works with leading biotech and pharma organizations to understand their biggest problems and deliver strategic solutions. Mike acquired his PhD in chemistry from the University of Waterloo and has several years of experience in identifying, interpreting, and solving complex research problems in the biochemical and biophysical spaces. As Systems Integration Manager, he works cross-functionally to connect subsystems to connect unique capabilities and functional solutions into comprehensive solutions for scientists. And finally, Rob has 25 years of experience in the life sciences, reagents, and services industry. After obtaining his doctorate from the MD Anderson Cancer Center in 1995, he co-founded the gene targeting and drug discovery company Lexicon Pharmaceuticals, and has since held other positions for Serologicals Corporation, Stem Cell Sciences, Ray Biotech, and most recently holds the position of Chief Biological Officer at Sinobiological. He's published his research in major scientific journals, including Nature and Science, and has authored 2 college textbooks focused on the study of stem cells and nanotechnology. Thank you all for being here. We're very excited to have you presenting today. So at today's session, we have 4 primary objectives that we're going to be covering, but our ultimate goal is for you to walk away with an arsenal of resources and tools that will enable your next discovery. So by the end of this session, you'll learn how the versatility and flexibility of surface plasmon resonance makes it an ideal technology for a wide range of biomolecular applications, and how SPR studies can provide you with comprehensive characterization of key COVID-19 reagents to accelerate the lead time on any novel solutions. You'll also walk away with know-how on developing a highly selective and specific COVID-19 antigen test, and how the next generation of SPR platforms are fueling infectious disease research through enhanced label-free detection and biomolecular analysis. So to get us started, I'll now pass it over to Soleil, who will tell us more about the capabilities of surface plasmon resonance.
Thanks, Sajni, for the great introductions. Welcome, everyone. Before I jump into what SPR is, I'd actually like to provide a bit of background on the inspiration behind today's presentation. So despite the stark realities that have been imposed on us by the COVID-19 pandemic, we've continued to observe the unrelenting determination of the scientific community to bring novel solutions to market. And bringing novel solutions to market efficiently continues to depend on having access to reliable, versatile, and sensitive research technologies to understand the SARS-CoV-2 virus and keep advancing life-changing discoveries. So that's our mission at Nicoya, actually, is to improve human life by helping scientists, like many of you on this call, succeed. And we empower scientists all over the world with accessible solutions that deliver high-quality data. And since inception, our goal has been to democratize access to a powerful technique known as surface plasmon resonance, or SPR. And we design and build cutting-edge SPR solutions that are accessible to every scientist. And we take a really user-centered approach to ensure that you get the data that you need. And so what exactly is SPR? In short, SPR is an analytical technique for studying molecular interactions. It's label-free and it provides results in real time. So you're actually able to observe binding events as they occur and derive data from the association and dissociation phases of an interaction. Unlike conventional or more traditional techniques that only provide you with yes/no data or more qualitative data, SPR actually allows you to measure both binding kinetics and affinity of an interaction, which gives you really detailed insights into binding strength and stability. So with SPR, you can investigate which molecules interact, why they interact, and how strongly they interact together. It's both real-time and label-free, which makes it a technique that is flexible and really information-rich for evaluating a wide range of biological molecules. And as a highly sensitive technology, SPR also allows us to distinguish between different time-resolved binding profiles of key interactions for a wide range of applications. So it's no wonder with all of these different features that SPR has been increasingly adopted over the last 25 years as a gold standard in biophysical analyses. And it's become prevalent in leading biotech and pharma companies, as well as top-tier universities. So SPR is proving to be essential when it comes to rapid characterization of biomolecules. From studying key biological pathways to developing novel biotherapeutics, it's a quick and informative way of selecting key candidates for accelerated product development. And again, compared to more commonly used binding techniques such as ELISAs, SPR assays provide you with deeper insights on your targets in up to half the time, and they automate several aspects of your workflow to reduce the experimental variability. So you save time in sample prep, which is always great. You no longer need to always have your purified samples, and you can actually eliminate a lot of the time-consuming labeling steps and a lot of the specialized reagents from protocols. You'll also increase your investigative abilities by reducing the amount of sample needed for full kinetic characterization. And so with the versatility and flexibility that we've described with SPR, it really is a Swiss Army knife technology that can offer comprehensive data for your applications, including, of course, SARS-CoV-2 research. So since the virus was identified over a year ago, a plethora of novel biotherapeutics, vaccines, and diagnostic tests have remarkably become available on the market. And that's thanks to, again, the dedicated researchers and their access to technologies like SPR. And one key area of development during this time has been antigen-based detection for rapid point-of-care testing. So we all know that we can't hold our breath for the widespread vaccination to become available. So we really do need to do the best that we can to curb the spread and minimize the impact of COVID-19 through rapid testing. If the virus becomes endemic, this will need to continue well into the future. So the primary method of detection has been PCR-based tests. So this is a gold standard for sensitivity, but there are problems with it. So one is that these tests can often produce a positive result long after a patient is no longer infectious. And in addition to that drawback, they also unfortunately require specialized facilities and technical staff for processing. And that just sort of undermines the ability to mass test. But luckily, there's an alternative to PCR. And it's a method of diagnostic testing that overcomes the bottleneck by directly testing for the specific viral antigens. And viral antigens are just proteins on the surface of the virus that we can detect. So these simple antigen tests can produce results more rapidly, making them easier to deploy in point of care settings. And our approach to developing a COVID-19 antigen test was greatly accelerated by the comprehensive insights that SPR provided at every step. The pandemic is obviously a prime example of when having access to the right tools can really move the dial in terms of getting novel solutions to market quickly and effectively. In the next section, we're going to talk about one of the critical requirements, in addition to having the right tools, that need to be met as we develop the assay. And this is really centered around the right reagents. So every researcher on this call knows that you can't really embark on a project like this without having the utmost confidence in the quality and consistency of the reagents that you use. And we, of course, were fortunate to collaborate with Sinobiological for really high-performing antibodies and target proteins. So I'm going to pass it back to Sajani now, who will introduce our next speaker.
Thank you very much, Soleil. So now I'd like to invite Rob to share a bit about Sinobiological's response to the pandemic and how they ensured that researchers studying SARS-CoV-2 were equipped with the best and robust reagents.
Thank you, Sajani and Soleil, for that very nice introduction and handoff. And I just want to take a second to thank all of the attendees for our co-hosted webinar today. We really appreciate your presence. As Soleil mentioned just a few slides ago, PCR-based detection of nucleic acids for COVID-19 or really any infectious disease just is not optimal for rapid results or for point of care diagnostics. If you want rapid results and if you want to be able to test in the field, you do have to test for the antigen. And that's where Sinobiological comes in, especially with respect to SARS-CoV-2 and COVID-19 related testing. As of January 11th of last year, as soon as the sequence for COVID-19 was published, literally 11 days later, Sinobiological produced and launched the world's first recombinant binding domain for the spike protein. And literally one day later had the first antibody available for binding to that protein. Since then, Sino has been the worldwide leader in providing the various mutants for the spike protein, as well as the nucleocapsid protein, and a variety of monoclonal, polyclonal, and even recombinant antibodies, which bind specifically to these proteins and other proteins related to COVID-19. Again, Sino has the world's largest selection of recombinant antigens and correspondingly the antibodies that bind to those antigens for COVID-19. These products are utilized to promote basic research, certainly to promote the development of vaccines and therapeutics for COVID-19. And then, as in this relationship and this partnership with Nicoya, to develop next generation immunodiagnostics assays that are rapid and can be used in the field at point of care sites. And I also want to mention very briefly that Sino, in fact, does have the world's largest selection of full-length biologically active recombinant proteins. So it's not just focused on infectious disease or even COVID-19, but really the world's largest selection of proteins for basic research, diagnostics, and therapeutics development. Next slide. Thank you. In relation to COVID-19 and the spike protein, this slide is actually a bit out of date. Sinobiological now has upwards of about 80 variants of the spike protein. These include the very famous and well-known D to 614G spike variant, the UK version, the UK variant, the Brazilian variant, and as well as the one from South Africa, all of which have been shown to be more infectious than the original variant of COVID-19, and some of which may, in fact, even be more deadly. When Sinobiological provides these spike proteins and the corresponding variants, we always make sure that they're biologically active, and we always provide biologically active data. For example, flow cytometry data, which will assess the affinity of the variant to its binding to the ACE2 receptor and compare that to the wild type original spike protein. So some of the graphs that you can see in here are some of the variants that are being compared to the wild type for their affinity for the ACE2 receptor. And you can see that some of them have an even higher affinity for the ACE2 receptor, and some of them have a lower affinity for the ACE2 receptor. And I want to mention that later on in this webinar, my colleague Mike at Nicoya is going to talk about several of these spike protein receptor binding domain variants and how they have been validated along with cytobiological antibodies in the Nicoya SPR system. Next slide, please. And I believe this is my last slide, and this is just a good example of assay validation using surface plasmon resonance technology in a partnership between Nicoya and Cytobiological. So on the left side of the slide, you can see various types of monoclonal antibodies which have been validated using the SPR system developed by Nicoya, as well as several spike proteins that have been tagged and also validated in the system, tagged either with HIS or with an FC tag. The detection and binding kinetics were analyzed for SARS-CoV-2 antibodies binding to these particular catalog numbers of spike proteins. And what you can see here now is raw data that's been extracted from the open SPR-XT surface plasmon resonance analytical instrumentation and analyzed by trace drawing software to look at one-on-one kinetic binding affinities. This was done at, if I remember the concentrations correctly, 150 nanomolar, roughly 50 nanomolar, 16 nanomolar, 5.6 and 1.85 nanomolar of antibody for either buffer or in the presence of 50% serum. And what you can see here is the association constants as well as the dissociation constants in comparison between that for buffer and that in 50% serum are almost identical. And this is roughly at nanomolar concentrations. We can see very quick saturation and binding between the SARS-CoV-2 monoclonal antibodies with the various versions of the spike protein. I don't have this data, but I do want to mention it as well, that the Nicoya team also utilized an antibody that binds specifically to the FC tag, which was linked to the receptor binding domain of the spike protein. And when they did this, they enhanced the signal intensity by roughly 300%. And this allowed the detection, lower limit of detection, to reduce from nanomolar to picomolar concentrations of spike protein. And this is well within the range of what would occur in a human biological setting in vivo. So I just want to congratulate my colleagues at Nicoya for a job well done. We appreciate you utilizing our reagents and validating our reagents in your system. And we look forward to a really productive partnership with you. Thank you.
Yeah, thank you, Rob. I know that it's really exciting to see how quickly Cyno is able to sort of churn out new variants and new things for us to test. And we have seen, throughout this presentation, and Mike will go into it in a little bit more detail of how we kind of take that into account, but definitely keep throwing new antibodies and variants our way, and we'll keep testing them. Looking forward to it. So really just taking a step back for people who might not be as familiar with these types of tests. So how do these antigen tests generally work? So an antigen test is based on an immunoassay that captures the antigen of interest via a high affinity antibody, which you can see in that middle graphic there. And it indicates a positive test through a reporter system, which is the image on the right. So in this case of SARS-CoV-2, the spike protein is a suitable antigen. And that's due to its presence on the surface of the virus and also its lower sequence similarity within the coronavirus family. So for capture and detection of the antigen, two antibodies are typically used that bind with high affinity and specificity to different epitopes of the spike protein, forming an antibody pair. So because the antigen tests are generally less sensitive than PCR, it's really, really critical that you get the highest performing antibodies and detector molecules, and you really carefully select them to maximize the limit of detection of the assay itself. Yeah, and so there are several key characteristics to look for if you yourself are taking on a project like this. And it will really define with these detector molecules how successful your assay will be. So some questions to ask yourself right off the bat is, what is the specificity of the antibody for its target? Another thing is, will it cross-react closely with related strains? Will it bind to the antigen with high affinity and stay bound for the duration of your assay? And will the resulting complex be stable enough to kind of withstand any washing steps or additional reagents that you might need to perform? So from the outset of the project, we knew actually that SPR was a tool that could provide answers to all of these questions. And not just sort of yes or no or more qualitative answers. We would have super detailed kinetic and affinity characterization of candidate biomolecules. And this could guide our selection process by profiling the entire interaction between the antibody and antigen and having association and dissociation constants. So this really allows for deeper investigation of leads and ultimately results in more confidence when it comes to selecting your final candidates. And so sort of splitting this up into a couple of different areas for assay optimization. One is really that high quality data. So with SPR, you can confidently achieve high quality and meaningful data throughout the development process. And this obviously underpins the whole basis of the technology and really is why it's such a powerful tool for any project. Additionally, by using SPR to select antibodies with a high degree of specificity for their target, you can ensure a really reliable and sensitive test that minimizes the possibility of false positive results during clinical diagnoses, which is definitely something you want to avoid. The third thing is about LOD. So meeting LOD requirements for diagnostic tests is another huge challenge that developers face, and it makes it more critical that you select the optimal antibodies. So SPR is a tool that can help us achieve a lower LOD, and this is by finding higher affinity antibodies via screening and ranking them based on their individual affinities. And after the ranking is done, we can actually bin them to ensure that there is no epitope overlap. And this allows you to add a secondary detector to your assay that is really effective. And finally, the fourth point, and anyone working in this field knows that new antibodies are always being added to the library, thanks to groups like Sino. As new discovery research is actually being done, it's really important that you have a tool that you can continuously sort of validate and improve the reagent selection that you have within your assay. So SPR provides you with a super quick and cost-effective method that drastically cuts down the amount of time it takes to adapt quickly and select the best detector molecules for your assay.
Thank you, Soleil, for breaking down the key components of the immunoassay and how SPR actually enables us to comprehensively analyze all the reagents for detector selection. So now to walk us through how to actually develop the COVID-19 test with optimal selectivity and specificity, I'll now pass it over to Mike.
Thank you, Sajni. Thank you everyone here for joining us today. So what I'm going to do now is I'm going to take us through how we detail how to select an optimal antibody pair for our SARS-CoV-2 detection system. So as has been mentioned a few times in this talk, the first step is essentially creating this assay is to select the primary antibody, or what are referred to as our capture antibody that has a high affinity for the target protein. So in this case, the target protein that was selected was the SARS-CoV-2 spike protein. So selecting this high affinity antibody is crucial as it will ensure that the assay can effectively capture, and as Soleil has mentioned, stay bound for a sufficient amount of time to our detector protein. For this to occur, we've sourced many commercially available options and actually sourced several of these monoclonal antibodies from Sinobiological that were raised against the SARS-CoV-2 spike protein. And we've tested these as potential primary antibody candidates. So using our benchtop platform, we performed a series of SPR experiments to first determine the kinetics and then the affinity of each antibody. And then these were compared and ranked against each other to select our optimum antibody for the assay. So for this to be performed, we immobilized the spike protein onto our sensor surface, and then each antibody was injected at different concentration ranges to then calculate the associated kinetics for them. All of these antibodies that were used exhibited a high affinity, which was in the low nanomolar to the picomolar range. And then with this time result binding profile, we were able to eliminate any of our lower affinity antibody candidates, speeding up our selection process. Now, the affinity of the antibodies are important, but we also have to ensure that these candidates don't cross-react with similar strains. So for this, each candidate underwent additional characterization to determine their cross-reactivity with other known coronavirus strains. So given the relative sequence conservation of the spike protein amongst the different strains, the specificity and accuracy of this diagnostic test depends on our primary antibody only capturing the spike protein of the SARS-CoV-2 strain. So for this, affinity testing with the SPR provided a quick and easy way of comparing our binding ability of each of the antibodies selected to 3 of the more common coronavirus strains. This mitigates the possibility of us incurring any false positives during our assay. For this, SPR experiments were performed on the same set of candidates to confirm that specificity only to the SARS-CoV-2. In these tests, each antibody was screened against the SARS-CoV-1 and MERS spike proteins to determine if any binding occurred. And then we mapped these results in the associated table here. As you can see, for the first 4 candidates, we observed no cross-reactivity, while the other 4 candidates had mild cross-reactivity and thus were eliminated from our selection process. So at this point here, we've now used SPR to elucidate both the specificity and selectivity of our antibodies and help narrow down our search for the primary antibody candidate. As mentioned a few times here by Soleil, we also want to improve the limit of detection. This is often necessary for a diagnostic test so that we can detect small amounts of the virus that are in a human host. So for this to be done, it's often necessary to incorporate a reporter system into the immunoassay, which will amplify the signal of our positive test. Now, this reporter system can take on many forms, but a typical one includes using a secondary antibody, which is coupled to an enzyme that will produce, say, a coulometric change when it's successfully bound to the target and then exposed to specialized reagents, as depicted in our figure here. So for this to happen, we have to establish a pair of antibodies that can simultaneously bind to the target, in this case, the spike protein. So for this to be done, we need to test for epitope overlap of each of these given antibodies so that we can pair them against the SARS-CoV-2 spike protein. And thankfully, the SPR provided a quick and easy way for us to set up these assays and test for this overlap through a process known as epitope binning. So to perform this competitive assay, one way to do it would be to allow both antibodies to simultaneously interact with the target antigen while measuring the resulting binding activity. For this, the secondary antibody has to be able to bind to the antigen while it is bound to the primary antibody in order to be considered a successful pair, as this would indicate that both of them have different and non-overlapping recognition sites. Now, in our case, the epitope binning was performed on several pairs of our candidates. For this test, the spike protein was bound to our primary antibody, and then a secondary antibody was injected. And if binding wasn't observed, this informed us that the 2 antibodies were bound to different epitopes. So in the example we've shown here, we have a response signal detected for our antibody 1 to the spike protein. And then after the addition of the antibody 3, we also detect a signal. So that showed that both of these are able to bind to the spike protein, indicating that they don't have epitope overlap. Now, after performing each assay, we've mapped these results to a table to allow for visualization of the overlap. As we can see in this table here, the green boxes indicate that the pairs do not have competing binding sites, and thus we were able to derive which antibodies could be used as pairs for the assay. As seen here, we didn't go through all of the antibodies. As mentioned, some of them got removed from the selection process after failing the previous criteria. Now, to facilitate the diagnostic assay, sometimes it might be necessary to have a stable and reliable way to immobilize that antibody to the surface of your device. In many cases, this would involve modification of the antibody. So a common immobilization strategy involves using a biotin streptavidin conjugation, which either requires purchasing a biotinylated antibody or biotinylating your antibody in-house. Now, it's generally more cost-effective to do this biotinylation yourself. So if that is the case, you would need a quick and easy way of validating that this biotinylation was A, successful, and B, did not impact the quality of your assay. This impact could be felt due to loss of affinity of the antibody or in a change of its ability to actually even bind to the antigen. Now, this is another area where the SPR can be used to confirm this. And this is done using a binding study that will confirm the biotinylation process has successfully resulted in an active biotin group on your antibody, and then also can be used to confirm that this biotinylation does not disrupt the performance of your assay. So for these types of tests, they can be done in multiple ways. One of the simpler ways that this can be done is by capturing your antigen, so in our case, the spike protein, onto your sensor surface, and then injecting your biotinylated antibody to confirm that the binding was not affected. So this can be seen in the figure shown here, with the black curve shows that our biotinylated antibody was still able to bind to the bound spike protein. Now, to confirm that the biotin is present on that antibody that's bound, you can then inject an aliquot of streptavidin, and if binding is observed, then that would suggest that the biotin is successful. So as we can see here from the blue line, we had a nice response from our streptavidin, indicating that in this case here, the antibody was successfully biotinylated and no binding was affected. Now, this can also be done in the reverse way to validate the system, which we've also performed, but for lack of showing additional time here, we have not shown the figure. Now, after confirming that binding was not affected, if you're concerned that the affinity of your binding is affected from this biotinylation, a further SPR assay can then be done to demonstrate that this has not impacted the affinity of this antibody-antigen interaction. So what we've done here is we've performed another study where we've compared the affinity and kinetics of both the pre and post biotinylated antibodies. So as you can see from the results for both our antibody 2 and antibody 3, and from the two curves that were shown here, we're able to conclude that the biotinylation did not impact the binding affinity of these antibodies greatly. So in summary from our work, we were able to characterize several anti-SARS-CoV-2 antibodies against their spike protein to rank them for A, affinity, B, cross-reactivity, C, perform epitope binning to successfully select a pair of antibodies. And then the last thing was to ensure that any modifications that we did not affect their binding ability or affinity to our spike protein. So with access to our SPR platforms, we were able to successfully characterize these several candidates and select an antibody pair in under 6 weeks. As the core of the assay, a carefully selected antibody pair greatly accelerated the overall development timeline of our diagnostic test. And given the number of steps that are involved in this, it's important that we have access to something that's quick, versatile, and simple, such as our open SPR platform, to optimize this selection process rather than resorting to more conventional and usually user-intensive techniques, such as an ELISA. SPR also allows us to quickly characterize new antibodies as they come onto the market so that we can constantly be substituting higher performing antibody pairs into the assay to improve its performance and sensitivity. Now, as Rob alluded to on his slide there about the different spike variants, as new mutants and variants of this virus are being discovered, we're thankful for these reagent suppliers such as Simon Biological that continue to develop these recombinant proteins that carry these mutations. And with this SPR platform, it also enables us to move as quickly as they do to confirm that these antibodies that are selected for our assay are still able to recognize and bind with high affinity to these new variants. So as you can see from the assay results depicted here, as well as our table, we were able to quickly classify that all of the components of our assay are still able to bind with high affinity to some of these variants that are carrying these mutations. So what all of this shows us is that as scientists, it's important that we can stay nimble and flexible. And for that, it's imperative that we have access to the best tools and reagents for adapting quickly during these times of crisis. And I'll pass it back to you, Sajni.
Thank you, Mike, for walking us through how that immunoassay was actually developed. It was very insightful. So now I'll pass it back to Soleil, who will present on the next generation of SPR technology that is advancing infectious disease research.
Yeah, thanks, Sajni. And I think that, hearing from both Mike and Rob, you just see how mutually beneficial and valuable our partnership is. So it's super exciting to hear from both of you and see those sort of commonalities. But as we sort of look at the next generation, I think it's important that, we just once again say that at Nicoya, our goal is really to equip researchers, like many people on this call, with platforms that expedite your discoveries. And while SPR is clearly a super powerful technology, as we've seen today, it's our responsibility to recognize and act upon opportunities to improve it even further. And one of the first areas that we wanted to revolutionize was the fluidic systems that power SPR platforms, aka the status quo. So this finding that we had, combined with the feedback from the close relationships that we share with researchers around the world, we developed the world's first ever digital high throughput SPR platform. And this is Alto. So Alto, again, is the first digital SPR instrument. And what makes Alto digital? It's the first SPR instrument to be powered by a powerful liquid handling technology called digital microfluidics, or DMF. And this provides you with the ultimate freedom and flexibility in your research. And it really centers around the unique benefits that are enabled by this fluid handling technology. And so DMF is really at the core of Alto's benefits and what it differentiates it from traditional SPR instruments. It's an advanced fluid handling technology, and they're sort of a cross-section of how it works, but it replaces all the pumps, tubes, and valves of a traditional SPR instrument that can cause some problems during these types of projects. So it has complete on-chip fluid control, and the samples are sandwiched between two plates, and individual nano-sized droplets are moved across a voltage-controlled grid. And most importantly, DMF enables you to precisely control all of the fluidic operations necessary to perform highly robust SPR experiments. And as an additional nice thing, automated serial dilutions, which, no one's a huge fan of serial dilution, so anything that can take that off your hands is pretty important. And so the application of DMF to SPR really challenges that status quo. So Alto combines the power of nanotechnology-based sensors, digital microfluidics, and automation for enhanced biomolecular characterization. So it has 16 independently addressable channels. So it enables you to analyze up to hundreds of samples using up to 500 times less sample. So you get a lot of high throughput and you get to use a lot less sample than leading SPR instruments. In fact, as you can see on the slide, from just 2 microliters droplet of sample, you're able to derive a full set of binding curves for a complete kinetic and affinity analysis. And of course, it's crude sample compatible, so you don't have to waste time on sample preparation. And so Alto allows, DMF powered SPR allows for really precise and automated fluid handling operations to be performed on extremely small sample volumes. Like I mentioned, the automated serial dilutions, buffer switching, and flexible assay designs across all 16 channels. And so with performance metrics like instantaneous reactions and switching times, flexible assay designs, Alto really makes sure that you don't have to sacrifice the quality of your data or your time when you're ready to scale up your experiments. And Alto is really a sample in, answer out platform. You just walk up, load your samples in like you would a well plate, and the system performs the SPR experiment for you. And there's a short video of the droplet on the sensor and how data is acquired. So just to sort of summarize a lot of the things that we've spoken about today, but Alto specifically, with the integration of DMF and SPR, users are no longer limited by fluidics related performance issues for high throughput operations. And Alto provides researchers with one of the most reliable, user-friendly, and intuitive platforms for any workflow involving biomolecular characterization. And it also enables you to scale up throughput capabilities for large fleet libraries. So you can effectively screen and characterize in a single step with the lowest ever sample consumption. And by cutting down the time and cost of biomolecular characterization, Alto will also help you accelerate the time that you spend in R&D and early discovery, and that brings your novel solution to the market faster than ever. So our example today of developing a rapid antigen test is just one of the many that not only benefited from SPR analysis, but could be further optimized through Alto's capabilities. So among the many lessons that we've learned from the COVID-19 pandemic, continual innovation in analytical technology stands out as one of the most critical. And now is the time to set the precedent for accessible next generation platforms that will drive the novel discoveries of tomorrow and better prepare us for whatever challenges that might hold.
Thank you, Soleil, Mike, and Rob. That concludes our presentation today. We really hope that everyone enjoyed learning about the excellent research and development initiatives that our two organizers here are addressing when it comes to combating the COVID-19 crisis. And we really appreciate the time that you spent in listening to our presentation today. So we're going to transition to our quick Q&A session. So if you do have any last minute questions, please drop them in the Q&A box at the bottom of your screen. And if you do have any questions after today that didn't get answered or would like to learn more about any of the products that were mentioned today, you can get in touch through the contact information that you see on the screen here. So that takes us to our Q&A. I do see a couple coming in. So let's start with this first one here. I think this one is for you, Mike. So I believe it is referring to one of the previous data slides, but the question was from Tommaso. Do the sensorgrams shown in the latter, which we've kind of moved away from that, refer to a situation in which a washing step follows the bicoupling step? So I can go back to possibly.
Yeah, I think that was the biotinylation, the slide with the biotinylation work. Now, I'm assuming I guess I can't really give an answer, but I'm assuming it refers to during the actual biotinylating of the antibody. So after we biotinylate the antibody, we did follow the proper washing, and so it was washed previously to putting on to in the assay. So that would be, yeah, it's a situation where it's a washing step. A washing step was done, and then we did the assay on the instrument. I hope that successfully answers that question.
Yeah, thank you, Mike. We have a question from Ina saying, thank you very much for the interesting seminar. Your results are very impressive. Thank you, Ina. The question is, have you ever considered combining SPR with other biosensing platforms, such as electrical biosensing using FETs?
I don't know, Soleil, you want to jump in there? I've never tried using those other biosensing platforms, so I don't really have any experience with that, but it would be something to check out for sure, because we're always trying to, to see what we can couple our technology with to improve the scientific field. So that might be something that we would investigate further.
Yeah, absolutely. I would just echo that we have a seemingly insatiable appetite for combining different technologies. So we'll add that to the list, although I'm not personally familiar with that, but please feel free to email us because we'll look into it.
Awesome, thank you. So we have a question from Hannah, and the question is, why would you recommend SPR over other techniques like BLI? So kind of along the same lines, but why did we kind of go with SPR for this assay?
You want to jump in there, Soleil?
Yeah, sure. I think I can take the first whack at this and then follow up after. So they're both label-free techniques, so they do share similarities. But really for SPR, it is considered a more sensitive technique in terms of being able to detect exactly what's going on at the sensor surface. And the second thing is that with SPR, there is no, and with Alto specifically, and I saw another question regarding this a little bit. So maybe we can sort of get a 2-for-1 here, is that there is not a risk with DMF for evaporation. So because the samples are sandwiched between two plates, even though it's a really small sample where you would usually be concerned about that evaporating, we don't actually have to worry about that. And that includes throughout the assay. So you can see that interaction for an extended period of time and not have to worry about that affecting the concentration of your sample.
Great. Yes, that does answer the second question. I'm glad that was a 2-for-1 there. So Tiago, feel free to add a secondary question if that wasn't enough. A question from Richard: which optical detection convention do you use in ALTO?
Mike, do you want to take that optical detection?
Yeah, I'm a little bit, I guess, confused with the question, but I mean, it's very similar to what we would use in the open SPR. I mean, without going into some of the proprietary steps, but it's essentially like the similar. We're looking at the absorbance change on the sensor surface that we're detecting with the open SPR. It's also being done in the ALTO.
Yeah, so that's one thing that's helpful maybe here is that it's localized surface plasmon resonance because we do use nanoparticles on the sensor, and those are the same for ALTO and open SPR.
I think that does kind of tie into the next question, and then actually the secondary question to that is the new sensor setup in ALTO still similar to that of open SPR? So now kind of talking about the sensors themselves.
Yep.
Yeah. So yeah, I don't know if you. I mean, I can just jump in. So I think it's the original question was laser, not later, but yeah, the setup for the light source and the detection is very similar to the open SPR.
Cool. Our next question is: how accurate is your SPR system in measuring KD of low molecular weight targets?
That's a very good question. So that relies on a few factors. So one would be, it's not so much the low molecular weight, it would be the difference in molecular weight between the ligand and the analyte. So we are able to detect, analytes on the size like Daltons, but it depends on that molecular weight ratio between the ligand and the analyte, and then some other factors like how much ligand can we get on the surface, what is the KD for that interaction, how fast is the off rate. But if all those things are satisfied, then it's about as accurate as you would get on the vehicle or open SPR.
Great. We are cutting to the end of our time. So if you all are okay with it, I will pose one more question. So what is the limitation of kinetics that open SPR can determine? And can the software handle the situation of flowing the antibody over antigen?
Okay, so the second part, can the software handle the situation of flowing antibody? Yes, that's how the instrument works. So we could do it either way. Like in our case here, we put the antigen down on the surface, and then we flow different concentrations of the antibody over the surface to determine the binding kinetics. Now, the limitations of the kinetics, that would depend on like the system itself. So typically we can detect accurately a KD in the neighborhood of high micromolar down to low picomolar, but we are limited by the off rate of the interaction. And also, if you're trying to get accurate kinetics, we're limited by how fast the off rate is. So with our fitting software, we can determine full kinetics to get the affinity, or we can do affinity fitting. So typically you would switch to the affinity fitting software if the off rate is too quick to accurately determine the off rate for that interaction. I hope that satisfies the question.
Yeah, I think so. I just add that in case the question, like you can reverse the binding partners. SPR is really amenable to that. So.
Excellent. Well, thank you everybody for sending in those questions. Whichever questions we did not get to, we'll be sure to provide some answers to them in any follow-up emails after the webinar today. So thank you everyone for attending and sending in your questions, and thank you to our wonderful speakers for being here and presenting us with such an interesting topic. So that ends our presentation today. Definitely contact us if you have any further questions, and otherwise have a wonderful day, everybody. Thank you again.
Thank you. Bye.
Bye, everyone. Thanks for coming.
Thank you.